Good morning all of you. Today we are going to discuss one of the next topic, that is the genetic code. Before that we already discussed about the central dogma of protein synthesis. We know that is protein synthesis, in which what happened? Protein synthesis which is done by different two process, that is transcription and the translation. In that what happened? That is from the DNA, from one of the template strand of DNA, mRNA is formed, and mRNA means what? That is messenger RNA, and from this messenger RNA proteins are synthesized, in which from this DNA mRNA is formed. This process called as the transcription, and next one, from this mRNA proteins are synthesized, that is called as the translation. Okay. We know that is DNA, it is one of the genetic material which is present in the human being. It is responsible to express the different character, and this character which is controlled to that of the and regulate to that of the DNA. Second important thing is that in which the expression of the expression of a character takes place through the particular specific protein. जे कॅरेक्टर एक्सप्रेस होता है कुठून डीएनए पासून, इट इज कंट्रोल थ्रू दैट ऑफ द प्रोटीन. देन अनदर इम्पॉर्टंट थिंग फॉर दिस process of controlling of character, one of the codon which is responsible, means proteins are responsible. So these proteins which is made by different 20 amino acid. Okay, different 20 amino acid, and this different 20 amino acid which is made by different 64 codon. There are total 64 codon, and this 64 codon which is formed from different nucleotides, that is adenine, guanine, uracil and cytosine. These are the four nucleotides which is present in the mRNA, and these are the, we know that is different components which is responsible for the formation of DNA and the RNA. For the DNA, sugar molecule, phosphate molecule and one of the nitrogenous bases. Different nitrogenous bases, adenine, guanine, thymine and cytosine. These are the three components which is present inside the DNA, and in the RNA, that is sugar molecule, phosphate molecule and four type of nitrogenous bases, that is adenine, guanine, thymine replaced by the uracil and the cytosine. And through that of these four nucleotide, three codons which is formed, or specific codons which is formed, or 64 codons which is formed, and this 64 codon which is responsible to form the 20 amino acid. Out of this 64 codon, 61 codons are sense codon. त्याला काय म्हणतात ते? Sense codon, and three codon which is responsible to that of the termination codon. Means these three codon which is responsible to stop the protein synthesis. So that we are saying that out of 64, 61 codon, that is sense codon, and 3 codon which is called as the termination codon. And this termination codon UAG, UGA and UAA. These three are termination codon which is responsible to stop the protein synthesis. Okay. After that, in which 3 codon, or one of the terminology, that is triplet codon. Triplet codon, or what is meant by codon or code, means triplet codon means what? For the particular or the for specific amino acid, 3 nucleotides are responsible, that is called as the triplet codon. Which they are? That is, for example, AUG. It is in the 3 number. AUG which is responsible for the methionine. कशासाठी रिस्पॉन्सिबल आहे हा? मिथ्योनाइन. म्हणजे एखाद्या पर्टिक्युलर पर्सनचं जे नाव आहे ते एका पर्टिक्युलर स्पेलिंगने तयार होते, बरोबर आहे. तशाच प्रकारे इथे ट्रिपलेट कोडॉन जे आहे त्या पर्टिक्युलर अमायनो ॲसिडने किंवा न्यूक्लियोटाइडनी बनले असते. दॅट इज AUG विच इज रिस्पॉन्सिबल फॉर द मिथ्योनाइन. दॅट इज कॉल्ड एज द ट्रिपलेट कोडॉन, and it is responsible for particular amino acid. Okay. After that, so what are the different characteristics of genetic code? In that, the first is that genetic code is the triplet codon, means it is in the 3 number and it having responsible to particular amino acid. So that we are saying that genetic code is a triplet codon. 3 in a numbers, the codon which is in the 3 numbers, so that we are saying that triplet codon. Then the another characteristics of the genetic code, that is it has a particular or distinct polarity, means this codon which is present on one of the, for example, it is one of the strand of mRNA and it contains 5'3' direction. Okay, it having 5'3' direction, and on that numbers of triplet codons are present. For example, AUG, which is also called as the start codon. Okay, then we are saying that genetic code has a distinct polarity, means it will read by 5'3' direction. याला read करताना तुम्हाला फक्त 5'3' direction नीच read करता येईल, त्याला 3'5' direction नी read करता येणार नाही. ठीक है? So that we are called as a, the genetic code has a distinct polarity. Then the next characteristics, this genetic code has a non-overlapping. Means non-overlapping means, for example, it will not overlap. जसं आता AUG आहे, त्या AUGच्या बाजूला दुसऱ्या अमायनो ॲसिडचे स्पेसिफिक ट्रिपलेट कोडॉन प्रेझेंट असेल तर ते AUG तिथे प्रेझेंट राहणार नाही. फॉर एक्झाम्पल GUG आहे किंवा दिस GUG और ट्रिपल G विच इज रिस्पॉन्सिबल फॉर द ग्लायसिन. म्हणजे अशा प्रकारचे इथे आपल्याला वेगवेगळे अमायनो ॲसिड दिसणार. इट इज नॉट ओव्हरलॅप टू ईच अदर, सो दॅट वी आर सेइंग दॅट इट इज नॉट ओव्हरलॅपिंग इन अ नेचर. After that, the genetic code, it is commalase. Commalase means what? In which it is not having a gap. म्हणजे इथे कंटिन्युअसली तुम्हाला जे जेनेटिक कोड आहे ते कंटिन्युअसली mRNA स्ट्रँडवर दिसेल, ना की इथे आपल्याला गॅप दिसणार किंवा कॉमा नाही दिसणार. जसं AUG आहे, याच्यानंतर तुम्हाला कॉमा नाही दिसणार, त्याच्यानंतर GUG आहे, कंटिन्युअसली दिसेल, इथे कॉमा किंवा गॅप दिसणार नाही. सो दॅट वी आर सेइंग दॅट इट इज द कॉमलेस इन अ नेचर. आफ्टर दॅट it has a degeneracy. Degeneracy means what? That is in for the particular amino acid, 2 or more codon responsible means in which, for example, glycine. Okay, this glycine, it is one of the amino acid, and for this amino acid 2 or more codons are responsible. For example, triple G, GUG or G glycine, GUG, GAG, GCG. Okay, means these 4 triplet codon which is responsible for one of the glycine, so that it has a degenerate in a nature. After that, it has non-ambiguous. Non-ambiguous means what? Each codon for the particular amino, म्हणजे प्रत्येक कोडॉन जे आहे पर्टिक्युलर अमायनो ॲसिड साठी पर्टिक्युलर कोडॉन असणार आहे. ठीक आहे, पर्टिक्युलर अमायनो ॲसिड साठी पर्टिक्युलर कोडॉन असणार आहे. जसं आता आपण एक्झाम्पल बघितलं की ग्लायसिन जे आहे ते 4 टाईपचे आपल्याला इथे 4 टाईपचे कोडॉन दिसले, तर ते फक्त ग्लायसिन साठी असणार, ना की त्याला आपण मिथोनाइन म्हणू शकत किंवा दुसरे व्हॅलिन म्हणू शकत. ठीक आहे, तर अशा प्रकारचे जे आपल्याला ट्रिपलेट कोडॉन दिसेल, दॅट इज कॉल्ड एज अ नॉन ॲम्बिगियस इन अ नेचर. आफ्टर दॅट, initiation and termination codon. Initiation and termination codon means on the mRNA strand at the 5'3' direction, at the start codon means initiation codon means this codon which is responsible for the protein synthesis. At that time, at the 5' direction, one of the codon which is present and it is triplet in a nature and it is responsible for the specific amino acid and it is responsible for starting, means initiation means what? Starting codon, that is called as the AUG, means it is responsible for the starting of protein synthesis, so that it is called as the initiation codon. And termination codon, this termination codon which is responsible to terminate the protein synthesis, that is UAG, UAA and UGA. These are the three termination codon which is present at the 3' direction. Next, it is universal in nature, means it will not change. It is present same in all living organism. Then codon and anticodon, means this codon means which is present on the DNA and anticodon which is present on the tRNA. We know that there is RNA which contains the three non-genetic types, that is tRNA, mRNA and rRNA. Okay. Codon जे आहे which is present on the DNA strand and anticodon which is present on the tRNA and it is responsible for the protein synthesis. Okay, so all these characters which is seen into that of the genetic code. Thank you.